Okay, good morning. This is now your the start. We're going to start our lectures now on vertebrate fishes. Okay, we're starting with the Vertebrata philo group of chordata, and we're moving on to the fish. Okay, page 339 to 362 of your new textbooks. Okay, so just to start with the introduction, what do we have and what do we know about fishes? What is a fish? That is the first part of your introduction. What is the fish? So its ancestry, it has relationships with major groups. There are relationships between all groups of fishes, and we have these different components. So this is just a sort of a contents of what we're going to be going through. We're going to start going through with fishes, okay? Yeah, we're hoping to get through fishes by the end of next week already. So there's quite a lot to do in 6 lectures. It's quite a lot to do. We're going to look at the jawless fishes, the cyclostomata. And the cyclone. You know, if you think of a cyclone and tornadoes, in the shape of a cyclone or tornado, they're always circular. So that's what the cyclone means. It means circular. Stomata means mouth. So these are all the animals with circular mouths. And there are 2 animals that do not have jaws, 2 vertebrate fishes. First of all, what makes it a vertebrata? Remember that you've got the urochordata, you've got the cephalochordata, and then you've got vertebrata. So we talked about the tail chordates. We've spoken about the head chordates, cephalization. My tongue is tied today. I'm struggling. I don't know what it is today. And then in between, you've got vertebrata, between the head and the tail. Okay, vertebrates. So these vertebrates either use cartilage and later it becomes bone, hey? That forms this vertebrae that's flexible. And that allows them to move beyond the lateral flexion. We spoke about that. So the first group is the jawless fishes, cyclostomata, and we have 2. We're going to work with the hagfishes and the lampreys, okay? And we're going to discuss the differences. Then we move on to the chondrichthyes, which are the cartilaginous fishes, the fishes that are made up of cartilage. And there we're going to talk about sharks, skates, and rays, the elasmobranchii, and the chimeras, which is the holocephali. And those are the 2 different groups of cartilaginous fishes. Then we move on to the osteichthyes, the bony fishes. Those are the more characteristic common fishes which we know today. And there are 2 different groups. There's the ray fin fishes and the lobe fin fishes, Actinopterygii and Sarcopterygii. And the ray fin fishes are the most common fishes that we have around us today. Lobe fin fishes, we only really have 2 representations left. And lobe fin fishes are pretty special because one of them is the coelacanth. The coelacanth. Anybody ever heard of a coelacanth before? Coelacanth is a fish that was discovered by South Africans in the 60s. They were thought to be extinct for millions and millions of years, and they found them alive. Okay, we're going to talk about the coelacanth. And then the ray fin fishes are all the common bony fishes we have. Guys, this is a fish. Okay, their forms and their shapes and structures are amazing. The diversification and the diversity of different species is just absolutely amazing. And that's what we're going to try and get into. Then what we're going to talk about is we're going to look at some of the structural and functional adaptation of fishes. Remember we spoke about the chordates and the prochordata, that group. Now we're moving on to the vertebrata and the differences between their characteristics and what some of their morphological features, the characteristics allowed some of the animals to do. Okay? What's really special about the fishes is we're going to spend some time now with this higher organism and how it has an improved ability to move in water, locomotion in water, how it uses different functions and different structures to contribute to buoyancy, and that we're going to really focus on the swim bladder, as well as some other structures like the oils in its liver. They use the oils in its liver to contribute to buoyancy, these crazy things that these fish can do. Respiration, osmotic regulation, which is the ability to regulate salt in their body compared to salt in the water. So you have some fishes that have the ability to go from a marine, very salty environment into freshwater ecosystems, and they have the ability to regulate the salt content in their body and change their, and not be adversely affected by living in salt water and then moving into fresh water. Migration and homing properties or characteristics of species. And we're doing a lot of work on that in particular here because we have a lot of migratory species, but we don't know a lot about them. Reproduction. You're going to hear about the sneaky strategy in reproduction. Sneaky strategy. Some of the male fishes actually make themselves look like female fishes, and then they don't compete with other males. And when the males go and reproduce with a group of female fishes, the sneaky male's in the mix. And he produces with the females as well when they're relatively small. We're going to talk about that. And then a summary. So this is what we're going to work through in the next 2 weeks. Okay. Fish are amazing. And guys, fish are also so important. If you look at how much of the world is dependent on fish, it is the most important source of protein for people in the world. Fish. Okay, more people live off and require fish to contribute towards their nutritional intake than any other source of protein. Okay, fish. If we lose them, guys, and they're also one of the more vulnerable parts of the world. If you look at the different species that have got conservation status, the different groups, because they're in water ecosystems, and whatever you do here affects them down there. You cannot put a reserve fence around them and protect them. You have to protect ecosystems. You have to protect ecosystem processes. You know, when you talk about migration, you've got the most valuable commodity, animal commodity that is sold and processed between people in the world. Do you know it is an eel? It's eels. And some of these species that have migratory requirements between the marine environment and freshwater environments, they're exceptionally vulnerable to the way that we're managing and using our resources. You build a dam, you destroy the whole population of eels in one go. Build a new dam, your eels are gone. Do you understand? So the vulnerability of this group is also quite high. We are changing freshwater ecosystems with hydropower developments, with dam construction, with quality impacts. Remember, we spoke about one of the major drivers of chemical or of ecosystems is actually wastewater treatment works and the effect that they have on ecosystems, massive impacts. Some fish, unfortunately, as well. Do you know that there is a limit of how much tuna you guys can eat? Because tuna from the oceans are now being impacted on by heavy metals. They are starting to accumulate heavy metals. And now they're worried that people who have this craving and eat a lot of tuna are starting to get sick because of the amount of heavy metals that you get from fish as well. So there's some big concerns. This is a picture of the sardine run. So this is a population of sardines that migrate up the south coast of South Africa. And this annual event actually allows a massive population of sharks, dolphins, other fish species to occur. And it's finely tuned to how the different temperatures of currents change in the southern coast. When cold water is supposed to go up the warm coast, because of some of the storms and winter events, some of the cold water is shifted up towards the east coast. And in that cold water that's shifted up to the east coast, a lot of these fish are forced up the east coast. And as they go up the east coast, the cold water starts to disappear and they start to get trapped in this warm water because they don't want warm water. And they provide a phenomenal food source to a lot of other animals on the East Coast. And these activities, if you guys were aware in the news last week, there was a new article on how climate change is going to probably end this whole process. Okay, so you must see that they're being driven and what's being affected. Okay, fish are generally thought to be water-dwelling animals. But do you know that when they started coining the word fish, they included seals, whales, amphibians, crocodiles, and hippos. Even hippos were at some stage considered to be fish. So fish is a very dangerous word because you cannot refer to everything as a fish. Is a dolphin a fish? Yes. Some people say yes, some people say no. Dolphins are mammals, but we commonly refer to them sometimes as fish. So it's very difficult. Dolphins are not fish, they are mammals. So what is a fish? The question is then, what is a fish? So after the 1600s, when we started understanding that there was a difference between vertebrates and invertebrates, we started to also differentiate the different chordate groups and the more developed gnathostomata, the more jawed vertebrates apart. And we realized that we have reptiles, mammals, amphibians, and birds as well. Okay, so we needed to start packaging the differences. And we realized that the main fish group cannot include everything because there are some animals that belong to different groups. So we started to learn that you cannot just refer to everything as a fish. Whales are not fishes, they are mammals. Okay, so what happened? So today we started, and as we developed, we started to realize that we need to differentiate. So, by the way, dolphins don't have gills. Dolphins breathe air with their lungs, like whales, hey? They need to come up to the surface and they need to take a gulp of air, and in their lungs they hold their breath and they go underwater. So dolphins and whales do not have gills, okay? So one of the first things that we can use to differentiate fish is to say that all fish have gills. They have appendages, and most of them, the appendages are in the form of fins. Some fish rarely have lobe fins. They're not fins, they're not rays with fins like the lungfish. Does anybody in this class come from Mozambique? Anybody from Mozambique? In Mozambique, in the lowland rivers, if you go north of Maputo, there is a type of fish that goes and makes a hole in the mud. And then when the rivers dry up, it lives in the mud for many years. Okay? I'm going to show you in some of the next lectures what they look like. But the ladies walk around in the mud and they feel with their toes to find these little hard ridges that these lungfishes make. And then they dig them out. And they can take out a fish that's many kilograms big. They can dig them out the mud. Some people, when they make bricks, I'm going to show you the video, they make bricks and they don't realize that they've included these lungfish in the brick. And then they go make their houses. And after the first rains, the fish come out the walls. It's crazy. Those fish don't have fins. They have lobes. I'm going to show you. It's crazy. Be careful where you get your bricks. That's the take-home message. Okay, the skin of the fish. They have an ectoderm. They have a covering that is usually covered with scales, and it is of a dermal origin, but not all fish. You know the catfish, the barbet? We caught one last week. It's a fish that has a skin. It doesn't have scales. Okay, but it is not one taxonomic unit, the fish. So it's very dangerous if you just use the fish to represent that particular group. And as vertebrate biologists, okay, what you are learning in this module, it is very dangerous when you refer to taxonomic groups as the fish. Okay, this chapter is the chapter on fishes, but it's partly here to teach you that fish overlap between many different groups. So you've got to be very careful when you use the term fish. You've got to actually refer to the different phyla or subphyla. And I'm going to show you how to do that. Fish do not form one monophyletic group, which means that there are many different supposed ancestors that have resulted in differences between the fishes that we have today, and we generally put them all into one group. And because they have different theorized ancestry, you have resulting today different groups of fish that all look very similar, but they actually are very different morphologically. Okay, so it's very dangerous to use the common word fish. Remember, this is very similar to the way that you guys used to use the word vertebrates, because you used to learn that there were only invertebrates and vertebrates. But I've asked you to stop thinking of the word vertebrates and rather think of the word chordates. Chordata. You've got Urochordata, Cephalochordata, and then Vertebrata. So now you know exactly what Vertebrata is. So guys, I also want to warn you, be careful when you use the word fish as a group, because it includes tetrapods. So if you include the word fish in the ancestry of fish, you're actually including the ancestry of all of the mammals and birds and reptiles as well. Okay, I'm going to show you what that means in the next slide. Okay, so all non-tetrapod vertebrates. So how do we do this? How do we say then what is a fish? What is a fish? If it's very difficult to say if it's very difficult to define exactly what a fish is, maybe it's easier for us to define what a fish isn't. Do you understand what I've just said? Maybe it's hard for us to define exactly what a fish is because it includes amphibians, it includes reptiles, it includes birds. So that's pretty hard. Maybe it's a little bit easier for us to make sure and define exactly what a fish is not. Okay, so we use that a lot in science. As a part of the philosophy of science, which you guys will learn, later on in your DipCon, or even if you do advanced DipCon type courses, you start to talk about the philosophy of science. So in the philosophy of science, what we do today is we've agreed that it's very difficult for us to prove things correct. It's very difficult. But it is very easy for us to prove things wrong. Do you know what I mean by that? It's difficult for us to prove that it is correct, but it's relatively easy for us to prove it wrong. And the example that's always given is, how do you prove that the sky is blue? How do you prove it? In order to prove that the sky is blue, you've got to make sure that at no point in time it is not blue. It must always be blue. And you've got to measure it during the day, you've got to look at it during night, you've got to look at it during different seasons. You've got to actually collect years and years and years worth of data to say, yes, the sky is blue. Okay, it's hard to prove that it is correct. But if I want to disprove it, if I want to say that the sky is not blue, on one occasion, I need to know that the sky is not always blue. And I can say your theory is disproved. The sky is not blue. So when we come up with hypotheses to test, we often refer to them in the negative to try and disprove our theory, because you disprove it once and it's thrown, you can disprove the whole theory. But to prove that the theory is correct, you have to make sure that there is no variability. There's no chance of it ever being wrong. And that's hard to do in science. Okay, so for fishes, if I can use the same argument, for fishes, it's very difficult to prove exactly what a fish is because it includes the other tetrapods, for example. So let's look at what fish are not. And we can reach consensus that all non-tetrapod vertebrata can be considered to be fish. Okay, so let me show you. Okay, so this is the tetrapods. So here, if you look at this cladogram, you can see here that we've included the tetrapods, which also includes later on the birds, reptiles, and mammals, okay? They come from here. And you can see that because the fish and the tetrapods are all related, they have got commonality at some point. Because of that relationship, you can't say that everything is fish, because it includes these tetrapods. So what we've easily said is, how do you then figure out what a fish is? We say, if you take these out, then everything else is a fish. So all vertebrates, all vertebrata, excluding tetrapods, are fish. Does that make it easier? But if you look at what types of fish you get, you have Sarconopterygii, which is the lobe-fin fishes. You have Acanthopterygii, Actinopterygii, which is the bony fishes, the fin-rayed fishes. You have Chondrichthyes, which is the cartilaginous fishes. Cyclostomata, which is the non-jawed fishes. You see, so you have all these major groups of fishes, but not the tetrapods. Although the tetrapods, because of their morphology and their characteristics, appear to have a common ancestor. Make a bit of sense to you? Okay, so what is important for you to know from the cladogram is, can you read these cladograms? Okay, so cladograms always from the bottom up show you what types of characteristics are shared by the different groups. And at some point when you start to get the differences between the two, you say, one has got this, the other one has got that. And you separate the two from the cladograms. But using the way that we classify organisms, you see if you go from the top down, you can see these are all vertebrates. They're all vertebrata. Now you know that we started with the chordates and said you have urochordata, cephalochordata. For the rest of this module we're working with vertebrata. Only vertebrata. Within vertebrata you've got the agnatha, which are the jawless fishes, and you've got the gnathostomata, which are jawed fishes and vertebrate, and the tetrapods. So the gnathostomata are the jawed vertebrates, vertebrata, I'm saying vertebrates, although I should stick to vertebrata, and the tetrapods. Remember, and everything other than the tetrapods are fish. Okay, can I say that once more? So, we have already started with, in terms of the chordates. The chordates, we've got three groups. Urochordata, the tailed chordates. Cephalochordata, the cephalization, the development of your head, the chordates. Okay, we've discussed the lancets versus the tunicates. The different types of protochordata groups, hey? Do you remember that from last week? But now we're only focusing on vertebrata. So the green line on top shows that everything that we're dealing with now is all vertebrata. Okay, within the vertebrata, you have two major groups. The agnatha, which refers to no jaws, and the gnathostomata, which are all the jawed animals, all the jawed vertebrata. And that includes tetrapods. And the tetrapods are the amphibians, the reptiles, the birds. They're not here because we're going to get to that group. It comes from here. The whole tetrapod group is in this lineage, you see. And everything apart from this one are fish. Okay, good. So when we look at the fish, what do we have? We have cyclostomata. Cyclo means round mouth, no jaws, round mouth. If you've got a round mouth, you've got like a sucker disc, you don't have a jaw. Then these two are extinct. Do you remember we spoke about them last week? Actually, ostracoderms and the placoderms, because they've got bones. Remember, these are the two guys that had some bones, and they use the bones as hardy plates around them. Okay, but these groups are extinct. Then you've got the cartilaginous fishes, which are all the sharks and the chimerans. Okay? They don't have no bones. They've only got cartilage. Then you've got all the bony fishes. Oxti refers to bones. Remember, those guys have got bony plates, but they've got external bony plates outside. These guys have got internal endoskeletons. Endoskeletons made out of bone. So this is the bony group. This is the cartilaginous group. And these are the guys that have got, also they've got cartilage, especially in their adults only, in their adult life cycle, but they've got no jaws. What do you think? Are you kind of getting it? You need to learn it. Okay, and I like going from the top down because it's easier for you to just put the differences together. If I asked you to redraw, I could ask you to relabel the top to say, okay, which of these are vertebrata, and you should be able to show all of them. Which of these are cyclist or agnatha, jawless, and you should be able to show them. If I gave you the names, you understand that? I could give you these names. Alternatively, see, I'm never going to ask you guys for each of these. I'm not going to do that. That's too much for diploma, okay? But what I will do is I will say, group those that are all agnatha and group those that are all gnathostomata, or all the jawless group and the jawed group. I could ask you, which are the fish? It's everything except the tetrapods. I could also ask you, I would also come down one level and say, talk about cyclostomata. What are they? And you'd be able to say, they are the hagfishes and the lampreys. I could ask you to describe and discuss the chondrichthyes. What are they? I could ask you to describe and discuss the osteichthyes. And what are they? I even want to ask you the difference between Actinopterygiae, the rayfin fishes, versus the Sarcopterygiae fishes, which are the lungfish and the coelacanth. Just the differences. You see, that stuff is important. Okay. And if you consider the bottom, okay, this is more for you to understand this. Why are they ordered the way they are? Here you can look at the relationships. Distinct head, tripartite brain, specialized sense organs, one pair of semicircular canal, cranium, remember that was very important, well-developed pharyngeal skeleton. Okay, that's all of the vertebrata. All of them have that. This is what separates vertebrata from urochordata and cephalochordata. Okay. And then from here, if you want to look at the differences between the agnathans, their teeth formed from keratin, and the mineralized tissue in the dermis and the vertebrae themselves, hard vertebrae. When these guys are adults, they develop like a cartilaginous vertebrae, but they don't have it for their whole life cycles. That's why these two are separated. Then if you want to look at the difference between, so these ostracoderms and placoderms are extinct, so we're not too worried about them. We want to focus on what's living. Part of the second visceral's internal arch modified as supporting elements for the jaws with teeth. So that's where the jaws come in. Can you see here? So all of these are now the gnathostomata. And now you have the differences between cartilage and bone. And if you go with the bone group, you can see the differences between the lobes. So the ray fin fishes and the lobe fin fishes, and the lobe fin fishes include the tetrapods. And we're going to explain that because something like a coelacanth has even got the tetrapodal motion. Okay, for example. Yes.
So the double line are those who are extinct?
No. The actual extinct refers to those that are marked with these crosses. Those are the animals that are extinct. The lines that do not continue are those that are extinct. So these ostracoderms and placoderms, why are they important? They're important because when we're trying to understand the difference between cartilage and bone, these are very good examples from the fossil records to show us that they started to develop bones, but they were using more for exoskeletons. Okay, but they were developing bones. So there's a relationship here, but that's all we need to understand. They no longer exist. Okay, there are only samples of them from the fossil records. And the acanthodians we're going to get to when we're trying to look at the differences between cartilage fishes and bony fishes. Okay. You need to understand this, guys. Okay. The next thing is, remember when we use these, so the last two are related. Because of our ability to order animals the way we do, we use Linnaean nomenclature, the Cladistics, to help us differentiate what is the kingdom. The kingdom is animals, hey? All of the animals, including all the 5 kingdoms of animals. The phylum is Chordata. You know what that means now. The subphylum is Vertebrata. You know what that means. Now you need to understand that now we're starting to look at Actinopterygii. And if you look at Actinopterygii, you can see we're talking about this group of fishes here. You see? So the Actinopterygii is the suborder. No, the class, sorry. So the class is Actinopterygii. And there would be other classes. So these different layers are used to help us order organisms and life on Earth into different groups. And we can identify them using this classification system. So you've got Animalia, the kingdom, Chordata, Vertebrata, Actinopterygii, which is the ray fin fishes. Then these suborders, order, family, genus, and species is what we then use as vertebrate biologists to then order all of the animals within those groups together, all the different species. And this is what we need. So we use this information. Why? Because you guys are told, when you do your assessment, you're going to be working with Oreochromis placidus. And you need to understand, how does this genus and species, the Latin name for the species, fit into the bigger picture? And if you wanted to know, you could take your textbook, your fish book or whatever, and you could work out which group does it belong to. Like if I showed you the lungfish and I said, guys, here's a lungfish. Can you classify it? Then you can look in your books and say, I know what the species is, the genus, the family, the order, the suborder. The class is not Actinopterygii. The class for the lungfish is Sarcopterygii. See, you don't need to learn it, but I want to show you how we go from this structuring and order all the way down to your species, so that you know where the species name comes from. Not what the actual name is, but you know how it's ordered, how we structure this all with nomenclature, classification. Can you see there's a relationship between our classification system, which gives us this genus and species that we use every day, and how it all fits into how the morphologically and structurally they're all aligned. Okay, and that's why, as for animal studies, you need to understand vertebrates and you need to understand what you're working with so that you can be able to put all of this together yourselves. Okay, if you're a biologist, you may be working with one group. I work in this group, by the way, only that group. That's my group that I focus on. It's one of the fishes. Remember now, excluding tetrapods, there are many different species. I've got lots of colleagues who do a lot of work on the elasmobranchii. A lot of you guys can one day go work on sharks if you want to, but I work in this group. Okay, I've also got lots of colleagues who work with the coelacanths. There's lots of money from South Africa going to coelacanth work, for example. And then, of course, I have lots of colleagues at the university. You know, from Prof. Parker to the other doctors, a lot of them work on the tetrapods. Okay, but we say, all the tetrapods are just one group of the fish. Okay, you can get them stick. Okay, then you get these type of graphs. Okay, so these are phenographs, and these graphs are different because what they're trying to show you is how did they, during the different time. So by the way, guys, this Cenozoic period is where we are today. So the animals that are in this level here are the animals that are still alive today. And this shows you that there were, according to the fossil records, there were some groups of animals that diversified, and there were many of them at different periods. And you can see the different layers, but some of them have become extinct. So you don't get ostracoderms and placoderms anymore. Acanthiodons, you don't get them anymore. They're extinct, you see. But it shows you in relation to the others what were there and where they are. If you have this line, this thin line, it means that there are very, very few representatives of what we have today in the fossil record. So the difficulty we have is for the agnatha, the fishes with no jaws, we have a lot of records of the ostracoderms in the fossil records, especially in the earlier fossil records, but we have almost zero records of lampreys and hagfish. Okay, but remember, these guys have got bony plates on the outside, so their bony plates were fossilized. These guys have got nothing but keratinized teeth. Keratin is like your nails. So we don't expect there to be many of them anyway, because of what they're made out of. Does that make sense? Okay, but if you then go up and you say, we've got the agnatha, jawless fishes, and apart from the jawed fishes, you have the gnathostomata. And the theory is that there was a vertebrata or craniata. Remember, it refers to this cranium. We spoke about that last week, this cranium that protects your brain case. That is another name for vertebrata. So the vertebrata, there was probably a common ancestor that diversified into the jawless fishes and the jawed fishes. So this is just looking at the difference. The way that we looked at this from the top down, I'm doing that from here now. The same thing from the bottom. To show you that they're all vertebrates or vertebrata. Stop saying vertebrates. Vertebrata. There's agnatha, jawless fishes, and gnathostomata, jawed fishes. And with the jawless fishes, you've got ostracoderms and your lampreys and hagfishes. That's it. And they're alive today. With your jawed fishes, you've got placoderms, which are extinct, as well as your cartilaginous fishes. Okay, those are the cartilaginous fishes. Where here, if you go up here, you've got the actinopterygians, the acanthodians, which we're going to talk about when we get to the differences between cartilage and bone. But here, this is your bony fishes. And your bony fishes diversified. Okay? But we also know today that somewhere even before this, you started to get the break off of these early tetrapods. And these tetrapods today become the amphibians and the amniotes, which are your reptiles, your birds, and your mammals. So you can order this all together, guys. And that's why I want you to hopefully, when we go through this process, you can always come back to this, refer back to this. I wish that this lab was our lab that we always used for vertebrate diversity, so I could put posters up. So whenever you're talking about something, I can say, where are we? Where are we? Maybe would it be better if I printed out this for all of you? And then you guys could always go back to it and say, where am I? Then you know where you are. That's probably a good idea. I'll do that for you. All right, and what we're going to do now is we're going to start with this group, lampreys and hagfishes. Then we're going to move on to the chondrichthyes, the cartilaginous fishes. Then we're going to move on to the bony fishes. And then I'm going to start with these tetrapods, and I'm going to do some work with the amphibians. Okay? Then Dr. Lenny is going to take over, and he's going to do the rest of the amniotes. Okay? But you're always going to come back to this. Yeah, that's all I need from that, I think. I might start with this a little bit later. And then we're going to talk about things like this. This is a hagfish. You know, have you ever heard the word hag? People used to call their wives, you old hag. Now you know why it's such a bad thing to call your wife a hag. Because the name hag comes from this fish, a hagfish. Does it have bones? No. Does it have a jaw? No. It has a circular disc, cyclostomata, circular disc, with these keratinized, like your nail, hard teeth. It's freaking gross. What does it do? It uses that teeth, it's a scavenger, and it uses that mouth, and it can attach to things. Some of them are like parasites, and they can grab stuff, and they can mince it up, and then they can ingest it. Gross. These things can get big as well. Hey? And it's slimy, slimy, slimy. You can't hold it. Hey? Hagfishes. Okay, what time does the next one start? All right. I'm going to stop there, guys.